Welcome students. So today we are going to discuss about the next topic, that is the structure and function of chloroplast, mitochondria and endoplasmic reticulum. So basically all the plant cells have various types of cell organelles, we can say, suspended in the cytoplasm. For example, chloroplast, mitochondria, then endoplasmic reticulum, Golgi apparatus, lysosomes are there, then ribosomes are there, nucleus, etc. So these cell organelles are actively engaged in various types of activities in the cells. So each cells, we can say, organelles have it has a definite structure and has a very specific role to play in the cells. So one by one we are going to discuss the first one, that is the chloroplast one. So chloroplast are the master molecules in the process of photosynthesis. So they harvest the solar energy and they convert it into useful biological, or we can say chemical energy. So the chloroplasts are present only in the green plants, algae and some bacteria also. So they are microscopic structure, differ in size and shape. So the chloroplast of higher plants are dish shape and oval in structure, which we can say about 10 mule in length and about 2 to 4 mule in diameter. So in the structure you can see, so the chloroplast is a double membranous structure, that is the outer membrane and the inner membrane. So the space between the two membrane is known as a intermembrane space. Okay, so the first, that is the outer membrane, so it is a semi-porous and permeable to small molecules and ions which diffuse easily. And the inner membrane, it is form a border to stroma, so it regulates the passage of molecules and we can say in and out of the chloroplast. So the next, that is the intermembrane space. It is present in between two membranes, which is about 10 to 20 nanometers. Okay, then the chloroplast shows the matrix, or we can say stroma, in which grana are present. A single chloroplast may show presence of 50 or more grana. So the granum, each granum is made up of stacks of thylakoids within these space. We can say which is space is found, which is known as a thylakoid space. Okay, then the stroma, it is an alkaline aqueous fluid or amorphous matrix, which is we can say protein rich, distributed throughout the chloroplast. So the chloroplast DNA or the ribosomes and thylakoid systems, starch granules, many proteins and different enzymes of Calvin cycles of the photosynthesis are found floating in stroma. Okay, so the thylakoid system, it is a membranous sacs suspended in the stroma. So the chlorophyll green pigment is found in thylakoid 1. Okay, so the thylakoids are arranged in stacks known as a grana. So each granum contains about 10 to 20 thylakoids. Okay, so the grana and the stroma, lamellae is there again. So the lamellae which connect grana are known as a grana lamellae, while the lamellae present in the stroma are called as a stroma lamellae. Now what are the functions? So the main function of chloroplast is to synthesize carbohydrates during the process of photosynthesis. Okay, then the chloroplast is the process of in the process of photosynthesis absorb carbon dioxide from the atmosphere and helps to reduce air pollution. Again, during the process of photosynthesis which takes place in chloroplast, oxygen is released in the atmosphere, thus maintaining the concentration of oxygen in air. So it takes place in the grana only. So the chloroplast are the main molecules harvesting the light energy, that is sunlight, and converting it into useful biological chemical energy. During the process of photosynthesis, ATP, that is energy molecules, and glucose molecules are formed along with the NADPH, that is high energy molecules. So these reaction takes place in the photosynthesis 1 and the photosynthesis 2, which is present in the granum. It against, we can say, the green color of plants, leaves and some algal membrane is due to the chlorophyll pigments only. So those photolysis of water, which is presence into presence of sunlight forming of hydrogen ions and OH minus ions, which combines releasing the oxygen in the atmosphere. Next, that is the photophosphorylation takes place in grana by we can say addition of IP, that is inorganic phosphate, to ATP, which give rise to ATP and NADH1. So it has again two types, that is photophosphorylation has two parts, that is cyclic and the non-cyclic one. Okay, so these are the some functions of chloroplast. Next, the structure of mitochondria. Again, then mito means the thread one, chondria means the granules. So this term was first coined by Benda in 1898. So mitochondria was first observed by Richard Altman in 1894. Mitochondria are the most important cell organelles in all the plant, we can say animals also, except some bacteria and fungi. So these are we can say popularly known as a powerhouse of the cells. So they are closely associated with aerobic respiration, where glucose molecules are broken down to carbon dioxide and water in presence of oxygen by releasing energy in the form of ATP. Okay, so we can say again mitochondria usually found in cytoplasm, which are small granular or filamentous bodies. Even they are vary in size, shape and number from cell to cell. Okay, so the average length of the mitochondria is about 3 to 5 mule, and average diameter is about 0.5 to 1 mules. Okay, so the main structure of mitochondria consists of 5 different parts. The first, that is the outer membrane. So it is made of we can say phospholipid bilayer, which is about 60 to 75 angstroms, which is thick, containing integral protein known as a porins. Again, it is a primary transporter of nucleotides, ions and metabolites between the crystals and the intermembrane space. Next, the intermembrane space. So it is the space between the two membrane, which is known as a perimitochondrial space. So it is about 40 to 70 angstroms in width, filled with watery fluid. Next, the inner mitochondrial membrane. So the inner membrane of mitochondria is folded into cristia. So the membrane is permeable only to the oxygen, carbon dioxide and water. So the inner mitochondrial membrane contains protein that perform redox reactions in we can say oxidative phosphorylation. So on this membrane, ATP synthesis enzymes transport protein and several antipode systems are present. Again, the cristia is present. Cristias. So these are the folds of inner mitochondrial membrane for expanding its surface area, for enhancing its ability to produce more and more ATP molecules. It contains about 2.5 we can say to total proteins present in the 2/3 of the total protein present in the mitochondria. So these cristia produce mainly ATP molecules with the help of ATP synthesis enzymes. They also show presence of tRNA, mitochondrial DNA genomes. Okay, so the cristia is having thousands of small particles on them, which is stalked. These particles are with the base piece and the stalk we can say as a head piece. Okay, so the head, pedicle and the base piece. So these are the spaced of a distance of about 100 angstrom. The head piece is about 75 to 100 angstroms in diameter, and the stalk is about 50 angstrom in length. So these particles, we can say elementary particles, are involved in electron transport and oxidative phosphorylation. These are also known as we can say electron transfer particles. And now recently it is known as F1 particles. Now what are the functions of mitochondria? So basically it is known as the powerhouse of the cell. We can say the prominent functions of the mitochondria is to produce ATP, the cellular energy for all metabolic activities. Because of this mitochondria are known as powerhouse of the cells. So next, that is the cellular respiration. Mitochondria are the site of all types of cellular respiration. Example: glycolysis, oxidation of puric acid, citric acid cycle, and oxidative phosphorylation, during which complex glucose molecules are oxidized to carbon dioxide and water, which releasing a huge amount of energy in the form of ATP. Next, that is the electron transport system. So the respiratory, it is again the respiratory chain, we can say this. The crystia or inner membrane of the mitochondria and the F1 particles are having the respiratory chain, which is engaged in oxidative phosphorylation. It consists of a cyst A or cyst B or cyst C, etc. So the ATPs are synthesized during the transport of electron through this chain only. Transaminations. So in mitochondria, many times we can say glutamate, prolines, and arginine, during this it has been formed. Okay, due to the process of transamination. Similarly, we can say oxaloacetic acid is converted to aspartate and asparagine during the same process only. So all the amino acids produced are used by the cell in different processes as and when required. So protein synthesis. So the mitochondria have their own set of DNA which is used to synthesize protein only. Storage of calcium ions, it is again the next functions. In many plants, we can say plant cells, mitochondria helps in the storage of the calcium one. Again, this mitochondria through their activities play major role in transport of different types of signals between cells and different organelles, especially through the reactive oxygen species only. Again, the regulation of cellular metabolisms. Almost all the cellular or the we can say metabolic activities in the cells are regulated or controlled by mitochondria, and they play a vital role in the regulation of metabolic activities of the cells. And there are various other functions also, such as we can say programmed cells death, then hemosynthesize reactions, then steroid synthesis, then hormonal signals. Okay, these are the functions of mitochondria. Next, that is the endoplasmic reticulum. So again, this endoplasmic reticulum or ER, was first observed by Garnier in 1897. The term ER means, we can say endoplasmic reticulum, it is a network for fabrics of membranes. So this endoplasmic reticulum, like chloroplast and mitochondria, is very important cell organelle present in the eukaryotic cells of plants as well as in the animals also. However, it is absent in prokaryotic. So it is a large dynamic structure, consists of crystalia, tubules, vesicles. So this endoplasmic reticulum is distributed throughout the we can say cytoplasm, and on an average it occupies about 10% of the cell volumes. It has a different part, that is a crystalia, tubules, vesicles also. Then it is majorly divided into two types, that is a smooth endoplasmic reticulum and the rough endoplasmic reticulum. So in the smooth, when we say smooth, it is a granular form of endoplasmic reticulum. It is called as a smooth because its membranes are not covered with the ribosomes. It is a major part. So the smooth ER, we can say, generally very common in cells which synthesize non-protein substances like phospholipid, glycolipids and steroidal hormones. So we can say that smooth endoplasmic reticulum is less stable than the rough endoplasmic reticulum. Whereas the second, that is a rough endoplasmic reticulum, so it is also known as a granular ER or granular endoplasmic reticulum because its membranes are covered with full of ribosomes. Then again we can say this endoplasmic reticulum or the rough endoplasmic reticulums are extensively formed in the cells which are actively synthesizing proteins, and it is more stable one. So those ribosomes of this rough endoplasmic reticulums are constantly being bound and released from its surface. So the presence of larger rough endoplasmic reticulum is indicative of we can say metabolically active cells. So the binding site of ribosomes on rough endoplasmic reticulum is known as a translocon. Okay, so these are the endoplasmic reticulum basic structures. Next, what are the functions of endoplasmic reticulums of smooth one? The smooth one's major we can say carries out many functions other than protein synthesis. It synthesizes lipids, phospholipids and steroids. So it again acts as a storage organelles. It also carries the metabolisms of carbohydrates and deoxifications of natural metabolic products, alcohols and drugs. It regulates calcium ions concentration in the muscle cells. The process of glyconeogenesis is carried out by this smooth endoplasmic reticulum. So it serves as a transitional areas for transport vehicles. It also functions in carbohydrates and lipid biosynthesis. So these are the functions, few functions of smooth endoplasmic reticulum. Next, what are the functions of rough endoplasmic reticulums? So this rough endoplasmic, it is mainly concerned with the protein synthesis only. So the synthesized proteins are transported to the Golgi apparatus. Again, it plays an important role in the synthesis of different types of membranes. And now this rough endoplasmic reticulum produces many types of transmembrane proteins and lipids for the synthesis of membranes of cell organelles like lysosomes, secretory vesicles, Golgi apparatus and cell vacuoles. The it again synthesizes the membrane and secretory proteins. Now this rough endoplasmic reticulum it plays we can say in white blood cells it produces antibodies, and in pancreatic cells it produces insulin. So these are the major functions of endoplasmic reticulums. I hope you all must have understand this. Thank you very much.